Hello, the students who have joined. Can you just confirm in the chat box if my voice is audible and the presentation is visible? Please let me know if my voice is audible and clear and the presentation is visible. Okay, thank you so much. We will wait for others to join and then we will start today's meeting. So let us wait for 1 or 2 minutes, then we will start today's meeting. Okay, so it's 7 p.m. and I will like to start today's session. We will add more students when they will join eventually. So yeah, uh, so welcome to the students who have joined today's meet. My name is Moshumi Dev and I am a PMRF fellow from the Department of Chemistry and Chemical Biology, IIT ISM Dhanbad. I am the TA for this course, NPTEL course of Organic Chemistry in Biology and Drug Development. Today it is the week 9 live session of this course and we will discuss some 10 to 15 questions today regarding your week 9 course content. And this type of question will help you to understand the course better and it will also help you to do this week's due assignment. And I hope you will learn something about this course more better in today's session. I will like to start today's session now and if you have any doubt regarding to any question, please do write in the chat box at the end of the session. I will try to answer those query. Okay. So I think my voice and my presentation is clear. If you have any doubt or any problem hearing my voice or seeing the presentation, please also write in the chat box. Okay. So yeah. Okay. So the first question of today's session is the in the drug discovery process, pharmacokinetic study is done during option number A, basic research, option number B, early discovery, option number C, preclinical study, and option number D, clinical development. Okay. So I will first show you the answer of this question and then we will discuss about this drug discovery process and also the pharmacokinetic study. Okay. So the correct option is the preclinical study. So during the drug discovery process, in the preclinical study, the pharmacokinetic study is done. So this is the basic chart of the drug discovery and development process. So as you can see, in the first step, a disease target is selected. Okay, I will raise a pointer. Yeah. So at the first stage, the disease target is selected, for which we are developing the drug. Then the first step is the basic research. So what happens in this basic research? These are the few points of this basic research. The target identification, target validation, assay development, genomics and proteomics. Okay. So after the basic research is done, the next step of the drug discovery process is the early discovery. So what are the steps of this early discovery? These are the computational studies. the high throughput screening, identification of heat. Somebody wants to join. Okay. Yeah, we were in the early discovery. So what happens in the early discoveries? The steps of the early discovery are the computational studies, the high throughput screening, the identification of heat to lead and lead optimization. So these are the steps of the early discovery of the drug discovery and development process. Okay. Now the next step, it is the preclinical studies. So what happens during this preclinical studies? There are in vitro studies, in vivo studies. There is a pharmacokinetic studies, formulation and dose and dosage studies. So there are a very huge discussion about each and every points of this drug discovery process. So we will skip all that and we will only focus on this pharmacokinetics, which in the question it is asked the pharmacokinetic study is done during which process. Okay. Then after the preclinical, there is a clinical development and there is a FDA and post-market surveillance. Okay. So now, what is the pharmacokinetic study? So yeah, there are very important two terms coined during the drug discovery process. These are the pharmacokinetics and pharmacodynamics. And many people got confused with which one is pharmacokinetics and which one is pharmacodynamics. So basically, you need to remember that the pharmacokinetics is what our body does to an drug. Okay, what our body does to a drug, it is called the pharmacokinetics. And what the drug does to our body, it is called the pharmacodynamics. So, in pharmacokinetics, there are some steps, and which is popularly known as the ADMET. Okay, the ADMET, ADME and T. So what is this admitter stands for? It is related to the pharmacokinetics and it stands for the absorption, the distribution, the metabolism, the excretion and T for the toxicity. Okay. So when a dose of the drug is administered in our body, what happens? It is the drug concentration in systemic circulation and the drug concentration in the site of action. Then what happens? It is the first happens that the absorption administration of the drug and the absorption of the drug. Okay. Then the distribution of the drug. It happens in the all the tissues of our body and all the places by the blood and other bodily fluids. The distribution of the drug happens. Then what happens is the metabolism. The drug is metabolized or excreted. E stands for excretion. Okay. And what is T? T stands for the toxicity. The toxicity is produced by the drug. So these are the few points what is related to the pharmacokinetics. It is basically what our body is doing to a drug which is administered. And then what is the pharmacodynamics? It is what is the drug is doing to our body. So as you can see, there is pharmacological effect, there is a clinical response and toxicity, effectiveness. These all are related to the pharmacodynamics. Okay, so it is very important to remember these two terms and what is the pharmacokinetic, what is the pharmacodynamics, because it is very intricately related to your drug discovery process and medicinal chemistry also. Okay, so I think the first question is clear. If you have any doubt regarding any point of questions at any point of today's session, please write in the chat box. I will try to answer those queries at the end of today's session. Okay. Now I will move toward our next problem. Okay. So the problem number 2, it says that which of the following statements is true regarding Lipinski's rule? Option A, log p value less than 5. Option B, molecular MW or molecular weight greater than 500 Dalton. Option C, hydrogen bond donor greater than 10. And option D, hydrogen bond acceptor equal to 20. So please read this kind of true false questions because it is very sometimes it got very confusing if it is true false or correct incorrect kind of question. So it is related to the Lipinski's rule, and this rule is very important for a drug discovery, for a compound to show drug likeliness or drug-like property. This Lipinski rule has to be followed. Okay. So let's see the correct answer first, and then we will discuss what is Lipinski rule, what is these terms, and what is all about. Okay. So the correct answer, that is the true statements among these following regarding the Lipinski's rule, is option A, log p value less than 5. Okay. So what is the Lipinski's rule of 5? It is some few rules, and if these rules are followed, then the molecule will show the drug-like property. It is also called the rule for the drug likeliness. Okay, so this is actually it is one of the basic and the most preliminary points for a molecule drug discovery for for a disease and the molecule. It must or it should actually follow the Lipinski's rule. So what is the Lipinski's rule of 5? There should not be more than 5 hydrogen bond donors. Okay, not more than 10 hydrogen bond acceptors. So what is the hydrogen bond donor actually? Hydrogen bond donor, it stands for the molecules like amines, okay, like NH or OH. These kind of functionalities, these are called hydrogen bond donor. And what is the hydrogen bond acceptor? Basic, very common hydrogen bond acceptor is the ketone group. This oxygen, it is a hydrogen bond acceptor, okay. And these molecule, this amine functionality, alcoholic functionality, these are called the hydrogen bond donor. So there should not be more than 5 hydrogen bond donor and not more than 10 hydrogen bond acceptors. So, and the molecular weight should be less than 500 Dalton. Okay, so higher molecular weight molecules are not very good as a drug moiety, and the C log P or log P value should not be greater than 5. So this C log P or log P value stands for the lipophilicity. So what is the log P? Okay, so the P, it is called the partition coefficient. So it is a measurement of the drug water solubility. Partitioning means the drug will be divided in the parts between the water and oil layer. And for our body, this oil stands for the lipid bilayers of our body. Okay, you all know that there is a what in our body there is more than 70% of water, and other there is a lipid bilayer in our body. So how this drug is distributed in this water medium and the lipid medium of our body, it is known by the we can know it by the partition coefficient value. Okay, so the P, it is the partition coefficient. It is nothing other than the concentration of CO divided by Cw. And what is the CO and Cw? It is the concentration of the drug molecule in the octanol medium divided by concentration of the drug the molecule in the water medium. This octanol, it is stands for, it is actually give the value for the as a lipophilic or the lipophilic nature. So if the log P value, log P, then log, what will be the log P? The log P will be the log of this value. That is the log of concentration of CO divided by concentration of Cw. Okay. So if the log P value is greater than 2, then it is the lipophilic drug. And if it is less than 2, it is a hydrophilic drug. What is lipophilic? Lipophilic stands for lipid loving. Means the drug concentration will be higher in the lipid bilayer. And what is the hydrophilic meaning? It means that water loving. Means if it is hydrophilic drug, the drug distribution concentration of the drug will be greater in the water or the aqueous medium of our body. So this log P value only applied for neutral compound. Okay. Lower log P, low penetration to CNS, and higher log P, low water solubility, means not suitable for oral administration. That is, if our the drug has a very high log P, this is not suitable for oral administration. It has to be administered intravenously. Okay. So I said that Lipinski's rule give us the very molecules which follow the Lipinski's rule. They have a very good drug-like properties. Okay, but there are few exceptions of this rule. Okay, not all the molecule will showing the drug-like property will follow the Lipinski's rule, and the several exceptions of these rules are this paclitaxel. It is starting from for the drug molecule of the Taxol, which is the anti-cancer drug. There is ivermectin and vincitris. Okay, so these are few exception of the Lipinski's rule. So I think it is clear, and I will now move toward our problem number 3. Okay, so it is very important, this Lipinski's rule, and I will suggest please read this carefully, and because you have to apply it many times if you are going to research regarding drug or want to learn more about this drug discovery. Okay. So problem number 3, it says that the incorrect statement among the following is option A, lesser the value of ED50, the drug will be more potent. Option B, larger the therapeutic index, safer is the drug. Option C, therapeutic index doesn't depend on the LD50 value. And option D is lower the IC50 value, better is the drug molecule. So let's see the correct answer first, then we will discuss what is the therapeutic index, what is this ED50, LD50 or IC50 values. Okay. So The incorrect statement among the following is option C, therapeutic index does not depend on the LD50 value. So what is therapeutic index? The therapeutic index is the ratio between the dosage of a drug that causes the toxic or lethal effect and the dosage that causes a therapeutic effect. Okay. So what is the lethal effect or what is the therapeutic effect? Actually, the ED50 value, it is called the median effective dose, and it is the dose that produces the therapeutic effect in the 50% of the population. And the TD50 or LD50 value, it is the dose that is the toxic or lethal to 50% population. Okay. So what is the therapeutic index? It is the ratio between the TD50 or LD50 and the ED50 value. So in humans, it is said the toxic value or the TD50 value. In the animals, it is called the LD50 value or lethal dose. Okay, so this both are the same. The toxic and lethal dose. These two terms are just interchangeable in humans and animals. Okay, and what is the IC50 value? Actually, IC50 value is basically coined for the inhibitors molecule, the inhibitor molecules, the enzyme inhibitors molecule. So it is stands for the inhibitory concentration in the 50% population. Okay, so higher the larger the this therapeutic index value, the safer will be the drug molecule. So it is a very important criteria for a drug molecule and the drug discovery process. Okay, and I will just quickly check if it is okay. There is no problem in anything. So, okay, so now, I think it is clear what is a therapeutic index, and please remember these terms like ED50, LD50, IC50, what is the meanings, and these values are very important because when you are doing a drug research and you are just searching for a molecule which will show the property of a drug, these values, these terms, and the values of these criteria, these are very important values because these show how safer is your drug, how effective is your drug. Okay, so next, we will move toward our next problem. So the problem number 4 is asking that in which of the following process the protein 3D structure of the target is a must requirement. So option A is ligand-based drug discovery. Option B is structure-based drug discovery. Option C is fragment-based drug discovery. And option D is all of the above. Okay. So let's see what is the correct answer for this question, and then we will discuss this drug discovery process and what is the correct answer and why it is the correct answer. So the correct answer is the structure-based drug discovery. So in the structure-based drug discovery, the protein 3D structure of the target molecule is a must requirement. So there are several types of drug discovery. So these are the structure based, in which the receptor 3D structure is known and the generation of interaction sites for the ligand is important. Okay. So in ligand based drug discovery, what happens that the receptor 3D structure is unknown and the generation of the pseudo receptor or direct similarity search has to be done. In atom-based drug discovery, a seed atom is required and high number of generated structure with questionable chemical accessibility is also required. In fragment-based drug discovery, a seed fragment is required and lower the number of the generated structure with good chemical accessibility we can have from the fragment-based drug discovery. So among this, the structure-based drug discovery, it is very common, and in this, the protein 3D structure is a must requirement. So what is the workflow of a SBDD project? So the crystal structure of protein available from the literature. So first, the protein for which we are designing the drug, we need to know that protein 3D structure, and we can have it from a protein database. Okay, and then the high throughput virtual screening is done, which stands for the HTVS. Then the identification of heat molecules is happens, and then the synthesize and purchase the heats, assay the heats, identify the active compounds, and the lead optimization. So it is a basic workflow of the SBDD project. You don't need to remember or memorize each and everything. Just understand what is the flow and how it is done. Okay, you don't need to memorize the things, but just read it like a very in detail how it is happening, and you will find it is very interesting. So among these three or four, the structure based drug discovery and the fragment based drug discovery is very important, the ligand based also. Okay, so please remember and read this drug discoveries very carefully because these are very important. Okay. Again, I am saying if you have any doubt regarding to any question, any problem, please write in the chat box at the end of the session. I will discuss those doubts. Okay. So I think it is clear. Now I will move toward our next problem. Okay. If you lost connection in any point of the lecture, please don't worry, because I will share this PPT, this slides and lecture video of today at the end of the session. So problem number 5, it says that identify the correct statement about combinational chemistry. Option A, one molecule can be done in one time. Option B, it has high risks of failure. Option C, it was developed using solution phase synthesis. And option D, it creates an impact in lead discovery and optimization. So you have to identify the correct statement about combinational chemistry among these four. So let us see the correct answer first, then we will discuss about this combinational chemistry. Okay. So the correct answer is it creates an impact in the lead discovery and optimization. Okay. So actually, this combinational chemistry, it is a technique by which large number of different but structurally similar molecules are produced rapidly and submitted for pharmacological assay. This technique uses the same reaction condition with the same reaction vessels to produce large range of analogs, and it was invented in late 1980s and early 1990s to enable tasks to be applied to many molecules simultaneously. And what is a combinatorial library? The products produced from a combinatorial synthesis are known as combinational library. The libraries may be a collection for individual compounds or mixture of compounds. Screening of the components of the library for activity using high throughput screening techniques enables the development team to select suitable compounds. Okay, and it is done using actually the solid phase synthesis, and thousands of molecules can be done at a time, and it has a very lower risk of failure. Okay, so the combination of chemistry within drug design, what happens first, the therapeutic target is identified and the lead discovery. The next step is the lead discovery, and the third step is the lead optimization, and then the development of the candidate and the drug, we get the drug molecule. So it is a basic flowchart of a drug design. And the combinational chemistry can impact in the lead discovery and lead optimization, means that the second and third step of this drug design. Okay. And what is the difference between the conventional and combinational drug discovery? So in conventional drug discovery, one molecule at a time can be done. And the combinational, in case of combinational chemistry, many molecules at a time can be done. Okay. Then the next step is similar in both of cases, that is the make, purity and test. Okay. And in the conventional case, the first is the make, then purity, and then the test. And then combinational, first make, then test, and then the purity has to be checked. Okay. In conventional drug discovery, hundreds of the molecules can be done at the time, but in the combination, thousands of the molecule can be done in a month. Okay. So in conventional, the lead generation is slower, but in combination, it is faster. And but the conventional, it has a high risk of failure, but the combination, it has the low risk of failure. And the synergy is the lead identification. So in both conventional and combinational drug discovery, we have the lead the lead identification has to be done, and the combinational chemistry is superior to the conventional in because due to these reasons. And this basically it creates an impact in the lead discovery and optimization. Okay, so I hope it is clear what is the combinational chemistry and how it is done. Okay, so basically it is a kind of computer based drug discovery as a part. So I hope it is clear. And now we will move toward the problem number 6. So the problem number 6 is says that which of the following agent doesn't have any effect of its own. The options are agonist, antagonist, partial agonist, and inverse agonist. So these are basically the receptors. Okay. And let's see the first what is the correct answer, and then we will discuss each and every terms and what is all about. Okay. So the correct answer is option B, antagonist. Antagonist doesn't have any effect of its own. So these all are the receptors. So what is the receptor? It is defined as a macromolecule or binding site located on the surface or inside the effector cell that serves to recognize the signal molecule or drug and initiate the response to it, but itself has no other function. Okay, so the following terms are used in describing the drug receptor interaction. So basically the receptors are what in our body which interacts with the signal molecule and drug, and it initiates the response to it. Okay, but itself it does not have any function. Okay, so in the drug discovery, this receptor molecule and receptor identification is very important. So what is the agonist? Agonist is an agent which activates the receptor to produce an effect similar to that of the psychological signal molecule. Okay, so if a agent which is added, it activates the receptor in our body to produce similar effect that of the normal physiological signal molecule. It is called the agonist. What is the inverse agonist? It is an agent which activates the receptor to produce an effect opposite to direction of that of the agonist. So there are two terms, agonist and inverse agonist. Agonists produce the physiological effect same as the normal physiological signal molecule, but the inverse agonist, it produces the inverse effect of the agonist. Okay, and what is the antagonist then? An antagonist which prevents the action of an agonist on a receptor of the subsequent response but does not have any effect of its own. So the antagonist molecule, it does not have its effects of its own at all, but it will prevent the action of the agonist on the receptor. Okay. So what is the partial agonist? It is an agent which activates the receptor to produce submaximal effect but antagonizes the action of the full agonist. Okay. So the partial agonist is still an agonist, but it will not produce the whole value of a full agonist. It will just produce a submaximal effect. And what is the ligand? A ligand is any molecule which attaches selectively to particular receptors or sites. The term only indicates affinity or ability to bind without regard to functional change. Agonists and competitive antagonists are both ligands of the same receptor. Okay, so these are some important terms, and you have to. I will suggest that please remember these terms carefully. What is the agonist? What is the inverse agonist? Antagonist? Because it will in the subsequent weeks when we will discuss the drug discovery and the molecules which are the used as agonists and antagonists. You need to remember these terms to understand those things better. Okay. So I hope the problem number 6 is clear. What is the antagonist? What is the agonist? And again, I am saying if you have any doubt regarding any question, please write in the chat box. At the end of the session, I will discuss those doubts. Okay. Or at any point, if my voice is not clear or the presentation is not showing, please also do write in the chat box. So let's move toward the problem number 7 then. Okay. The problem number 7, it says that low concentration of neurotransmitter dopamine has been implicated in Parkinson's disease, and L-DOPA is the most widely used medicine to slow down this disease. Which of the following statements is incorrect? Option A, dopamine cannot cross blood-brain barrier. Option B, L-DOPA acts as a product. Option C, L-DOPA can cross blood-brain barrier. And option D, L-DOPA is first decarboxylated by dopamine decarboxylase before entering the brain. So, we all know Parkinson's disease is a very important disease, and L-DOPA is a very important drug regarding this Parkinson's disease, and it is the very most widely used medicine to slow down this disease. So, let's see the answer first, then we will discuss what is dopamine, how the L-DOPA works on the blood-brain barrier. Okay. So the incorrect statement among these four is the L-DOPA is first decarboxylated by dopamine decarboxylase before entering the brain. So this statement is incorrect. The first three statements, it is correct. Dopamine cannot cross the blood-brain barrier. L-DOPA acts as a product, and L-DOPA can cross blood-brain barrier. Okay. Okay, so as you can see, it is the structure of the L-DOPA, or this is called levodopa. Levo stands for the L, or we know the levorotatory and dextrorotatory, it is the levorota. So levodopa or L-DOPA, it is outside in the periphery of the blood-brain barrier. What happens? It is actually there are two enzymes which converts this first L-DOPA is converted to 3-methoxytyrosine by the enzyme COMT. The COMT stands for catechol O-methyl transferase. Okay. So what this catechol O-methyl transferase enzyme does? It basically methylates one hydroxyl group of this L-DOPA. Okay. As you can see, this OH is converted to the OMe by the COMT enzyme. And there is a AADC enzyme which stands for the amino acid decarboxylase enzyme. Okay, and what it does, it is it basically decarboxylates this amino acid. Okay, and this is the dopamine. So from L-dopa to dopamine, it is converted by the AADC enzyme, and this is basically the decarboxylation reaction. Okay, so this L-dopa, it can actually cross the blood-brain barrier. Okay, so in the blood-brain barrier in the CNS, this L-dopa is converted to dopamine by this AADC enzyme, and then there is an enzyme called it is a myob enzyme. So what it does actually, it actually deaminase this thing. Actually, it actually oxidizes this amine group to the carboxyl group. Okay, so this myob enzyme, it oxidizes the amine group to carboxylate group, and we get the DOPAC molecule. Okay, so this happens inside the blood-brain barrier. By this method, the L-dopa acts on the Parkinson's disease. Okay, so please remember what is L-dopa and how it is converted to dopamine, and it is by the AADC enzyme, and L-dopa can cross the blood-brain barrier, and what happens inside the blood-brain barrier. Okay, this is very important. The Parkinson's disease and L-dopa, these things are very important. Okay, so please read these things carefully, and it is very important for your in annual examinations also. So problem number 8, it says that the following structure is parathion. Okay, this structure which is given, it is for the molecule, it is called parathion, and the options regarding are, it is what is the parathion? Is it a medicine for the treatment of glaucoma, which is option A? Option B, it is an insecticide. Option C, it is an antidote to cholinesterase poisoning, and option D, it is a medicine for the treatment of Alzheimer's disease. Okay. So you need to identify the correct statement about this molecule of parathion. So let's see the correct answer, then discuss what is the parathion. So it is an insecticide. Okay. So this is the structure of parathion. There is a in para position there is a nitro group, and to para to it there is a phosphoester kind of moiety, and it is actually called parathion or methyl parathion or cotton poison. And it is a chemical pesticide that should be used only in open field to control insects. Most commonly, it is used for cotton, soybean, and vegetable fields. So it is a brownish liquid that turns milky white when mixed with water, and it can leave yellow stain on the areas where it has to be it has to be sprayed and smells like rotten egg. And it is a kind of cholinesterase inhibitor. It is one of. Okay. So it is actually important to know few structures and few names of the molecules, because this kind of question can always come to any point, because there are so many drug molecules and so many insecticides and all these diseases for antidotes, and you don't need to remember, but please remember few examples like parathion, malathion. These are the insecticides molecule. Okay. So this is the structure, and some time what can be happen that you are not given the name parathion, you have only given the structure, and you are asked to identify what is the molecule, what is the name, and what is it is used for what is. So these are few common things. You just remember of a few example of this common molecules. You don't need to remember everything of the every molecule. Okay, but few common molecule you need to remember. So this was a comparatively easy question. Now we will move toward our problem number 9. I hope you don't have any doubt regarding this problem. Again, I am saying if you have any doubt, please write in the chat box. Okay. So problem number 9, it says that which of the following statements regarding neurotransmitters is not true? And option A is these they are secreted by neuronal cells. Option B, they are chemical messengers. Option C, they basically traverse through blood to act on a distant target cells. Option D, they carry out a cascade of reactions. So you have to identify the correct statement, the incorrect statement regarding the neurotransmitters. So first, let's see what among which of the following statements is not true, and then we will discuss about neurotransmitters. Okay. So the correct answer is option number C. They basically traverse through the blood to act on a distant target cells. Okay. Now, what is a neurotransmitter? Neurotransmitters are the chemical substances released by electrical impulses into synaptic cleft from the synaptic vesicles of presynaptic membrane. It then diffuses to the postsynaptic membrane, binds to and activates the receptor present, leading to initiation of a new electrical signal or inhibition of postsynaptic neuron. These are few neurotransmitters which are very important in our body. There is adrenaline, which all we all know. It is called fight or flight, and it produced in stressful situation increases the heart rate and blood flow, leading to physical boost and heightened awareness. So there is a GABA. Okay, and it is used for the calming effect. It calms the firing nerves in the central nervous system. High levels of improve focus, low levels cause anxiety, and also contributes to motor signal and vision. There is a noradrenaline, and it is called for used for concentration. It affects the attention and responding actions in the brain, contracts the blood vessel and increase the blood flow. There is acetylcholine. It is used for learning, and it is involved in thought, learning, and memory. Activates muscle action of the body and also associated with attention and awakening. Dopamine, it is called the pleasure neurotransmitter, and it is gives us a feeling of pleasure, also addiction, movement, and motivation. People repeat behaviors that lead to dopamine release. Okay, so there is a glutamate, which is a memory neurotransmitter, most common neurotransmitter it is, and it is involved in learning and memory, and it regulates the development and creation of nerve contacts. So there is serotonin, it is for mood, and it contributes to the well-being and the happiness, helps in sleep cycle and digestive system regulation and affected by the exercise and light exposure. And there is a endorphins, and this is for euphoria. It is released during exercise, excitement, and producing well-being and euphoria, reducing pain. Okay, so these are few important neurotransmitter, and so there is a very common confusion between the hormones and neurotransmitters. So there are distinct differences between hormones and neurotransmitter, and it is important to remember those. So the hormones are released by the endocrine glands, but the neurotransmitter, these are released by the nerve cells. Hormones travels through the blood to act on distant target cell. Neurotransmitters travel across the synapse and act to nearby cells. The effects of the hormones can be slower to build but longer lasting. The effect of neurotransmitter can be fast to act but shorter lasting. The hormones are part of the endocrine system, and the neurotransmitter are the part of the nervous system. So it is very important to remember the differences between the hormone and neurotransmitter, because it is some people actually many people get confused between the hormones and neurotransmitter and think these are the same, which is absolutely wrong, because the hormones and neurotransmitter are very distinctly different. Okay, so I think I am clear about neurotransmitters and what is the difference between the hormones and neurotransmitter. And please remember few examples of the neurotransmitter. Okay. So now we will move toward our problem number 10. It says that which of the following is true regarding cholinergic receptor. Okay. Option A, the inner part of cell membrane is hydrophilic in nature. Option B, the cholinergic receptors are of three types. Option C, the nicotinic receptors activate the ligand-gated ion channel opening. And option D, acetylcholine esterase enhances the effect of acetylcholine. Okay, so let's see what is the cholinergic first. Let's see which of the following statement is true regarding the cholinergic receptor, and then we will discuss about it in detail. So the correct answer is the acetylcholine esterase enhances the effect of the acetylcholine. Okay. So yeah. So cholinergic receptors, which also acetylcholine receptor, or it is stand for this AChR. Okay. So these are of two types. First, it is muscarinic acetylcholine receptor, which is stand for MARCHR, MACHR, and there is a nicotinic acetylcholine receptor which is NACHR. And this nicotinic acetylcholine receptors, it is of two types. It is neuronal type of nicotinic receptor and muscular type of nicotinic receptor. So, the stimulation of all autonomic glandia, the stimulation of the voluntary muscles, and the secretion of adrenaline from adrenaline medulla, it is done by this cholinergic receptors. And this nicotinic receptor fall into three main classes: the muscle, ganglionic, and CNS type. These are typically ligand-gated ion channels, and the muscle receptors are confined to the skeletal neuromuscular junction. Ganglion receptors are responsible for transmission of the sympathetic and parasympathetic ganglia, and CNS type of receptors are widespread in the brain. So these are the type of the acetylcholine receptor, which is called like cholinergic receptors. Okay, so it is important to remember what is the cholinergic receptors and the types of the cholinergic receptors, and okay. So the correct statement was the acetylcholine esterase enhances the effect of the acetylcholine, and please remember this details and this classification of the cholinergic receptors. Okay, the problem number 11, it says that the adenylyl cyclase converts ATP to option A ADP, option B CAMP, option C GDP, and option D GTP. So adenylyl cyclase is a enzyme, and it is very important enzyme for the conversion of ATP, and it actually converts the ATP. The correct answer is B CAMP. CAMP stands for the cyclic adenine monophosphate. Okay. So this is the structure for the ATP. We all know this is a there is a adenine moiety here. This is a ribose sugar here, and there is a triphosphate. Okay, and for this adenylyl cyclase, what it does, it actually cyclize this, and it is converted this ATP to the CAMP, or the cyclic adenine monophosphate. Okay, so this diphosphate, it is this PPI is this diphosphate group is eliminated by this adenylyl cyclase, and there is a cyclization happen. And the cyclization happens here between this number 3 OH group of this ribose sugar and this first phosphate group of this triphosphate. Okay, and this cyclic AMP molecule is very important because it involves in many cell signaling of our body. So it is basically the flow chart how it is happening. As you can see, this is the adenylyl cyclase enzyme, and there is a alpha subpart and a GTP, and there is a ATP. It is converting into the CMPT by this adenylyl cyclase enzyme. Okay. So I think it is clear what this adenylyl cyclase and how it is converting the ATP to CMP. Remember this name of this enzyme and what it does. Okay. So problem number 12, it says that which of the following is an organophosphate antidote. Okay. So the options are option A, pralidoxin, option B, GABA, option C, ecothiopate, and option D, malathion. Okay, so in the previous slides, I said that it is important to remember few names of the few important molecules, like in the drug molecule. If it is a drug molecule, it is a pesticide, it is a neurotransmitter. This is important. So now you can see in this question, it is asked that the following is an organophosphate antidote. Somebody wants to join right now. Okay. Okay. So that's why I said that it is important to remember the names and the functions of few molecules. Like as in the previous slide, I discussed the parathiol. So from in among these four options, you know that the option D, malathion, is an insecticide, right? Because I said that malathion, parathion, these are very common insecticides. So it is not the correct answer, option D. So among these four, GABA is a neurotransmitter, as I said. So what is the organophosphate antidote? The correct option is the pralidoxin. Okay, so this is the structure of of the pralidoxin molecule, and this is there is a pyridinium ion, and there is a oxygen present in this ortho position, and how it is happened. Actually, this is the antidote for organophosphate antidote. Actually, what happens that there is a acetylcholine esterase. As in the previous slide I discussed the acetylcholine esterase, and it is actually inhibited by this serine gas. Okay, and it is a toxic gas, and it is inhibits this acetylcholine esterase. And basically after inhibition, what happens that this acetylcholine esterase, it is getting deactivated. So when we use this pralidoxime molecule, okay, what happens? Like this pralidoxime molecule, first this H group it is eliminated and we get this O minus. Okay, after the elimination of this proton from this molecule, we get this O minus, and this O minus attacks this phosphate molecule. And what happens? This after the attack of this phosphate molecule, this acetylcholine enzyme is liberated. Okay, so we get the liberated liberated acetyl coenzyme acid sorry the acetylcholine esterase, and there is a oxime inhibitor complex is formed. So basically this molecule is used for the when there is a poisoning of this serine, and this poisoning affects the acetylcholine esterase molecule, and after this drug molecule pralidoxime, it is the antidote for this poisoning, and it basically binds to the this molecule, this phosphate, and this oxime inhibitor complex is formed, and we get the liberated acetylcholine esterase, which is now can be used in the body. So please remember the structure of pralidoxime and the mechanism of this molecule. Okay, so this is basically very easy organic reaction. As you can see, this O minus is acting attacking this phosphorus, and how it is happening I will just show you in the slide. So after this O minus is there is a liberation of H plus from this molecule, and if the H plus molecule is liberated, we getting this O minus, and this O minus will attack to this phosphate, and this oxygen will be liberated, and when it is there, we will get just like PO minus, and there will be O here with this N and this molecule, and we have O, and then acetylcholine esterase molecule, and when this negative charge is coming back to form a double bond with the phosphorus, this molecule is liberated and we get this molecule. Okay, so you don't need to remember the detailed mechanism, although it is very easy to remember because it is very easy mechanism, and I hope it is clear. If you have any doubt at any point of this question, please write in the chat box. At the end of the session I will discuss those doubts. Okay, now we will move toward our next question. So the problem number 13, it says that which of the following is the correct sequence for the adrenaline formation in the body. Adrenaline is the very important neurotransmitter, as I discussed in the previous slides, and it is formed by the adrenaline glands of our body, which is just situated upon the kidney. So the first option is L-tyrosine converted to dopamine, which is converted to L-dopa, and then noradrenaline, and then adrenaline. Okay. Option B, it is the L-tyrosine. It is converted to L-dopa, which is converted to dopamine, then dopamine to noradrenaline, and noradrenaline to adrenaline. Option C, the L-dopa is converted to L-tyrosine, then the L-tyrosine converted to dopamine, and the dopamine converted to noradrenaline, which is converted to the adrenaline. And option D, it is the L-tyrosine, which is converted to L-dopa, the L-dopa to noradrenaline, noradrenaline to dopamine, and then to the adrenaline. So you need to identify which is the correct sequence for the adrenaline formation in our body. First, I will show you the answer. Then we will discuss the adrenaline formation in the body, the sequences. So the correct sequence is the option B, L-tyrosine. L-tyrosine is a very important amino acid, which is then it is converted to the L-dopa. L-dopa molecule is again converted to dopamine, and the dopamine is converted to noradrenaline, and again last at the noradrenaline converted to adrenaline molecule. So this is the structure of the each molecules and how this is converted. As you can see, this is the structure of L-tyrosine. We all know tyrosine is a very important aromatic amino acid which have a CH2, then ortho2, then NH2 and COOH, and there is a phenolic OH at the para position of this group. And this L-tyrosine molecule, first it is converted to L-dopa. L-DOPA stands for L,3,4 dihydroxyphenylalanine. Okay. So what happens in the L-DOPA and this tyrosine to L-DOPA conversion? It is happened by the tyrosine hydroxylase enzyme. Okay. What this tyrosine hydroxylase enzyme does, it basically adds one hydroxyl group, this hydroxyl group, to the ortho position of this OH group which is present in the tyrosine. So we have a catechol kind of molecule here in the L-DOPA. Then in the next step, this L-DOPA, it is converted to dopamine, and the enzyme used is DOPA decarboxylase. So what this DOPA decarboxylase is, it is basically decarboxylates this L-DOPA molecule, and it actually eliminates the COO. Okay, the carbon dioxide elimination happens, and we get this amine, and it is called the dopamine. Dopamine is a very important neurotransmitter. Okay, as I discussed, dopamine is a very important neurotransmitter, which is associated with this pleasure feel or the calming feeling of our body. Okay, then this dopamine is converted to noradrenaline by this enzyme of dopamine beta hydroxylase. So what this dopamine beta hydroxylase enzyme is doing, it basically adding one hydroxyl group to the beta position of this amine. So this is the amine group, the carbon atom which is the amine is attached, it is called the alpha carbon. Okay, and this is the beta carbon. And what this dopamine beta hydroxylase does, it adds on hydroxyl group to this beta position of the amine group. Okay, and last but not the least, this noradrenaline is converted to the adrenaline, and the enzyme used is there is a actually the phenyl ethanoamine is used here, and N-methyl transferase enzyme is used. So what this N-methyl transferase molecule does, it basically transfers one methyl molecule to this amine group, and we get this adrenaline molecule. Okay. So I will suggest you to please not only remember the sequence for the adrenaline formation in our body, but also remember the enzymes involved in each and every step. Okay, like in the first step, the tyrosine hydroxylase enzyme, which is hydroxylating the tyrosine to the L-DOPA, which is adding one hydroxyl group to the ortho position of this tyrosinic hydroxyl group. Then the second enzyme involved here is the DOPA decarboxylase enzyme. Okay. Then the third, this dopamine beta hydroxylase, and the fourth one is the N-methyl transferase. So this type of question, the question can become like this, like in this from this adrenaline formation. It is a very important topic. Okay, so questions can be come from this what is the role of the tyrosine hydroxylase, okay, or what is the role of the dopamine beta hydroxylase. So this roles of these enzymes are very important, okay, and these enzymes, so I will suggest you not only remember the sequence but also remember the roles of these enzymes, and also please remember the structure of this molecule. So see, these are the very easy structures to remember. If you remember the structures easily, you can also remember the function of the enzymes, because as you can see, there is a the tyrosine hydroxylase addition of one hydroxyl group only happening here. Decarboxylase, only the elimination of carbon dioxide is happening from this molecule to this molecule. The beta hydroxylase, the addition of again one hydroxyl group is happening, and methyl transferase, the transfer of methyl group is happening. So from this adrenaline formation, remember the correct order or correct sequence of adrenaline formation, the names and the structures of the molecules of L, like L-tyrosine, L-DOPA, dopamine, and noradrenaline and adrenaline, and the enzymes involved in the each and every steps for this adrenaline formation. Okay. So I think it is clear how the adrenaline is getting formed and what is the role of each and every enzyme, and how it is happening. Okay. Now quickly I will move toward our next problem. So the problem number 14, it says that which of the following is false regarding the renin-angiotensin system? Option A, aldosterone is a steroidal hormone. Option B, vasoconstriction is caused by the action of angiotensin 1. Option C, aldosterone enhances the retention of sodium and water in the blood. And option D, bradykinin is destroyed by angiotensin converting enzyme. So renin-angiotensin system, it is very important system, one of the very important systems in our body. Okay, this is associated with the blood pressure actually. the hypertension and the blood pressure is also all that is happening with the renin angiotensin system. It's a very important system, and you should know the systems very in detail. So the correct statement, the correct answer means the first statement regarding the renin angiotensin system is number B. The vasoconstriction is caused by the action of angiotensin 1. So this is the renin-angiotensin-aldosterone pathway. So as you can see, this is starting the dehydration, sodium deficiency or hemorrhage, and then the decrease in the blood volume. Then the number 3 step is the decrease in the blood pressure. So number 4 is the uh, juxtaglomerular cells of the kidneys and the increase of the renin. So when the dehydration happens, what happens? The renin increase of the renin is happening, and the liver, there is a in our liver there is angiotensinogen, and then what happens is increased renin and and this angiotensinogen it caused the increased angiotensin 1, which acts on the lungs. Okay, in our lungs there is a angiotensin converting enzyme which basically converts angiotensin 1 to angiotensin 2, and then this increased angiotensin 2 it goes through the it causes a vasoconstriction of the arterioles. Vasoconstriction means the increased pressure in the arterioles, and then blood pressure increases until it returns to the normal. Okay, and what happens, this angiotensin 2 it also acts on the adrenal cortex. Okay, and there increased aldosterone is also happening, and this increased aldosterone what happens that in kidneys increased sodium and water reabsorption and the increased blood volume. So vasoconstriction of the arterioles and the increased blood volume both increases the blood pressure until it is normal. So basically it gives us a flow chart what happens when our blood pressure is dropping, and it is caused by the sodium deficiency or hemorrhage or dehydration. And after that this renin angiotensin and aldosterone system it works and increases the blood pressure until it is returning to the normal. Okay, so there is this also this flowchart. There is adipose tissue, and from this hormones are these molecules are secreted. This aldosterone molecule is very important for the sodium retention and high blood pressure in the molecule. And what it does, the high blood there is these are the side effects of the high blood pressure: oxidative stress, damage to DNA, and vascular remodeling. Okay, there is the arterial stiffening or arteriosclerosis, renal fibrosis, and cerebral disease or stroke. And these are the arterial fibrillation. These are the side effects of high blood pressure. So this renin angiotensin aldosterone pathway is a beautiful system. How it is worked, you should know how it works, and what is the role of renin and angiotensin 1 and angiotensin 2. And I will suggest please read this system carefully in details, because in for your in the examination or your understanding the course better, this system is very important. Okay, so now I will move toward our last question of today's session, and it is says that the correct statement amongst the following regarding the antihypertensive drug enalapril is: it is an irreversible inhibitor of angiotensin converting enzyme. It is a option B, it is a product. Option C, it got poor bioavailability, and option C, it is a transition state analog inhibitor of angiotensin converting enzyme. Okay. So what is the correct statement about the regarding the antihypertensive drug enalapril? It is the option B, it is a prodrug. So this is the structure of this enalapril molecule. Okay. So what is NLRP? It is an angiotensin converting enzyme inhibitor. Is a prodrug. When hydrolyzed by esterases, it's active enalapril. Enalapril is the active enzyme, and it is hydrolyzed by esterases to its active form. With the mechanism of action, the enalapril competes with the angiotensin 1 for binding at the angiotensin converting enzyme, blocking the conversion of the angiotensin 1 to angiotensin 2. So angiotensin 2 is a vasoconstrictor and a negative feedback mediator for renin activity. Lower concentration result in decreased blood pressure. Enalapril may also act as a kinase 2, then it degrades the vasodilator bradykinin. So what is happening, I will tell you basically in the previous slide if I repeat. So as you can see, there is a when the blood pressure is lower, what happens that the angiotensin 1 the concentration of angiotensin 1 is increased, and this is converted to the angiotensin 2, by the angiotensin converting enzyme. So this angiotensin 2 works in various manners to increase our blood pressure by vasoconstriction of arterioles and increased blood volume. So what happens? The angiotensin 2 basically increasing our blood pressure. So what is the role of the antihypertensive drugs? They basically decrease your blood pressure. So for the high blood pressure patients, you see the antihypertensives are given. Okay, and there are very different kind of antihypertensives, like there are beta blockers, there are this and the, but one of the very important antihypertensive class of drugs are angiotensin converting enzyme inhibitors. So what this inhibitor does, they inhibit is this angiotensin converting enzyme, so the angiotensin 1 does not converts to the angiotensin 2. As you know, angiotensin 2, it basically increases the blood pressure. So if we can inhibit the production of the angiotensin 2, we can basically decrease the blood pressure, or we can basically manage the hypertensive disorder. So this NRPL molecule, it acts as a reversible inhibitor of this angiotensin converting enzyme. It basically binds to the angiotensin converting enzyme site, and in place of angiotensin 1, and blocks this site, so that the angiotensin 1 cannot be converted to angiotensin 2. Okay, so this is one class of this antihypertensive drug, and I will suggest, please remember, please note, read different types of antihypertensive drugs, like there is sodium, there is reabsorption for the sodium in our body, there are beta blockers, there are this angiotensin converting enzyme. Just read few. You don't need to remember the structure of these drugs. Okay, you don't need to remember. I just gave this structure to for your better understanding. But please read the classification of antihypertensive drugs and the examples from the each and every class. One or two examples of each and every class. So that is, thank you. This was the last question for today's session, and I hope I am clear about the questions we discussed, the topics we discussed today. And this was the week 9 session for your NPTEL class of the organic chemistry in biology and drug development. These slides and all what we discussed, and these slides I will just share with you in due time. Okay, and today's lecture video, it also be uploaded to the NPTEL website. And if you have any doubt, I will just share my, okay, I will just share my email ID with you. If you have any doubt, please write an email to me. So in the chat box, I have shared my email ID. If you have any doubt regarding today's session, or any thing which we discussed today, if you have any doubt, please write an email to me, and I will see you in the next class for the week 10 session of your course. Okay. So thank you for joining today's class. I hope it is clear what we discussed today. And if regarding anything you have any doubt, please write an email to me in this email ID. And I will meet you in the next session. So thank you all for joining today's class. And I will now close the session for today. Okay. Thank you so much.